Bipolar cells with ionotropic receptors respond to glutamate with depolarization, OFF bipolars. Bipolar cells with metabotropic receptors respond to glutamate with hyperpolarization, on bipolars. OFF bipolar. Depolarizes in dark. Hyperpolarize in light. On bipolar. Depolarizes in light. Hyperpolarize in dark. 2. Horizontal cells. Transmits signals horizontally in OPL from rods and cones to bipolar cells. Main function: to enhance visual contrast by causing lateral inhibitions. Are antagonistic interneurons, inhibit photoreceptors by releasing GABA. Glutamate from cones and rod goes to horizontal cells and then releases GABA back into rods and cones. Provide inhibitory feedback to photoreceptors and inhibitory feed forward to bipolars. 3. Amacrine cell. Heavy checkmark receive information at synapse of bipolar cell axon and ganglion cell dendrites. Cone amacrines mediate antagonistic interaction among on a bipolar, off bipolar and ganglion cell. Heavy checkmark rod amacrine receive input of rod bipolar and deliver to on and off bipolar ganglion cell. Thus, rod signals undergo additional synaptic delay. Amacrine cells help in temporal summation and in initial analysis of visual signals before leaving retina. 4. Muller cells. Non-neural, glial cells. Extends from inner segment of photoreceptor to ILM. Functions IPL. 1. Plays a supportive role. 2. Buffers ionic concentration in extracellular space. 3. Forms ELM and seals subretinal space. 4. Glycogen metabolism. 5. Maintains clarity of vitreous via phagocytosis. 6. Neurotransmitter uptake and conversion. Applied. Activation of Muller cell in neuronal degeneration in retina. Increase growth factor secretion. Long-lasting gliosis may lead to subretinal fibrosis following RD and ERM formation leading to RD. Ganglion cell layer. 1.12 to 2.22 million ganglion cells. Second-order neuron of visual pathway. The electrical response of bipolar cells after modification by amacrine cells is transmitted to the ganglion cells, which in turn transmits their signal by means of action potential to brain. 2 types. 1. On-center cells, excited by the light in the center of their receptive field. 2. Off-center cells, inhibited. Other subgroups of retinal ganglion cells, tonic cells driven by L or M cones, foveal, for high visual acuity. Tonic cells driven by S cones. Extrafoveal, for color contrast. Plus phasic cell. Concentrated in fovea, faster conducting than. Ganglion cells important in movement detection. W ganglion cells. 40%, small receive most of excitation impulse from rods. Essential for scotopic vision. X ganglion cells, 55%, medium size. Responsible for color vision. Y ganglion cells. 5%, largest. Responds to rapid movement or change in light intensity.